to do these kinds of problems. So when you go to solve a genetics problem, you always need to write down all the information that you're given and everything that you can deduce. So it's kind of like those word problems, I guess you did back in middle school or something. I can't remember now, but the word problems that we all hated. So it's that same kind of thing where you write down everything you're given and everything you know. So in the first problem, we're working with peas. So in peas, the allele for smooth seed coat, big S, is dominant to the allele for wrinkled seed coat, little s. Give the expected genotypic and phenotypic frequencies for each of the following crosses. So when they ask you for a frequency, when they're asking you for a number, you can express that differently, right? You can do it as a decimal, a fraction, a percentage. They're all good. So we have a heterozygous times a heterozygous. Yes, it's a white screen because I'm going to be writing on it. Right? So I need a white screen so I can write. So our first cross is one that you already know without having to do anything. A heterozygous times a heterozygous. What kind of cross is that? Where we cross two heterozygotes together. What kind of cross is this called? A monohybrid, right? So we already know the answer to this, but as I said, write out what you know. So what we know is that big S is dominant to little s. So big S is for the smooth coat, little s is for the wrinkled coat. So because it's dominant, we know that big S, and big S, little s are all going to be smooth, right? Because the big S allele is dominant. And therefore you need two little s's in order to be wrinkled, right? So that we know. So our first cross is a monohybrid cross. Even without doing a cross, you should be able to tell me what the phenotypic and genotypic ratios are, because it's a monohybrid cross. So we already know the answer to that. So what's the phenotypic ratio? For a monohybrid cross? It's 3:1. It's 3:1 smooth right to wrinkled. So that we know. And the genotypic frequency or ratio is what? Right, because we already learned this. It's 1 to 2 to 1. Right, because we know, right, so those are the genotypes of the gametes produced. We get big S, big S, big S, little s, big S, little s, and little s, little s. So 3 of the 4 offspring are going to be smooth. 1 is going to be wrinkled, a 3:1 phenotypic ratio. And we say big S occurs once, big S, little s occurs twice, and little s occurs once. So a 1 to 2 to 1 genotypic ratio. In B, it says a heterozygous with a wrinkled individual. So we know that a heterozygote is going to be big S, little s, and the wrinkled has to be little s, little s. So here we have a 1-to-1 ratio. It's a 1-to-1 or 50-50 or 0.5 and 0.5 or 1/2 and 1/2 of smooth to wrinkled, right? Half the offspring going to be smooth, half the offspring will be wrinkled. When they ask you for the genotypic frequency, again, it's 1-to-1 or 50-50 or 1/2 and 1/2. So they're the same in that case. And C was to cross a heterozygous with a homozygous smooth. So we're going to cross a heterozygous with a homozygous smooth. Right? So here we see an example where we can simplify our Punnett square. Right? Because we know that one of the individuals is going to have gametes big S and little s. But the other one, all the gametes are big S, right? So you could do big S, and do, but why? This one is just going to be a duplicate, column, so we don't even need it, right? So we got big S, and big S, little s. Right, so again, our phenotypic ratio is 1-to-1, or I'm sorry, is 100%. It's 100% smooth, right? Because everything has a big S. Our genotypic ratio is 1-to-1. Right, big S to big S, little s. So half the offspring are homozygous dominant, and the other half are heterozygous. But I didn't really need to do, the second column because that just duplicates my effort. So I don't like to do any more work than I have to do. So sometimes you can simplify. So questions on those first ones? Okay, because I gotta erase this to move on to the next problem. So next problem. In peas, the allele for red flowers, big R, is dominant to its allele for white flowers, little r. And the allele for smooth C coat, big S, is dominant to the allele for wrinkled C coat, little s. So we already know the C coat. Now we have flowers. So determine the expected phenotypic frequencies from a cross between a plant heterozygous for both flower color and seed shape with a plant having white flowers and wrinkled seeds. Okay, the first thing we're going to do is write out what we know, right? So we know that big R is the allele for the red flower color. Little r is the allele for white flowers. We know that a big R is dominant to little r, so we know that if you're homozygous or heterozygous, you have red flowers. You have to be homozygous recessive to have white flowers. And then for the seed coat, we just did that. Smooth and a little less in order to be wrinkled. All right, so we wrote down all the stuff that we know. Now the cross that we're doing is an individual that's heterozygous for both flower color and seed shape. So heterozygous for flower color and seed shape, and we're going to cross that with an individual that has white flowers and wrinkled seeds. So in order to have white flowers, got to be little r, and wrinkled seeds, little s. So those are the two individuals, right, that we're crossing. So with this first individual, remember we can do our FOIL type thing, right, to figure out the genotypes of the gametes, right? So big R consort with big S, big R consort with little s, little r consort with big S, and little r consort with little s. So those are all the possible gametes from that one plant. Now the gametes from this other plant here, our white wrinkled plant, they're all going to be the same, right? There's only one type of gamete to be produced by this individual, right? Because they're homozygous recessive for both characteristics. So like I did before, we don't have to duplicate our effort, right? You don't have to do little r, little s, little r, little s, little r, little s, little r, little s, because it's all the same, right? So we're not going to bother with those because the ratios end up being exactly the same. Big R, little r, big S, little s. Big R, little s, little s. Little r, big S, little s. And little r, little s, right? So those are the combinations of all those alleles in the gametes. So there's 4 possible combinations. This individual has red flowers, smooth seed coat. This one, red flowers, wrinkled. This individual is white flowers, smooth. And this one is our white wrinkled. So it's an equal ratio. So both the phenotypic ratio and the genotypic ratio are the same in this case. 1 to 1 to 1 to 1. Oops, I missed my one there. Or each one is 25% or 0.25 or 1/4, right, for each genotype and for each phenotype. Because in this case, each phenotype is unique and each genotype is unique. And there's a 1 out of 4 chance. Now, if you had written out little r, little s 4 times, you'd still end up with the same ratios, with the same fractions, with the same percentages. It's just that you would have duplicated your effort. Well, not duplicated, you would have quadrupled your effort. So why do that? So does this problem make sense? I'm going to move on. So each screen gets wiped out when I move on. But this is being recorded, so everything is there. You can go back to it later to look at it. So our next problem has to do with cattle, right? With cows. You know, cows are female, bulls are male, so we just call them cattle. And they have different coat color patterns. And so let's write down what we're being told. So the red, and red is not red, it's a reddish brown color. So red big R, big R. And then we have white, which is big W, big W. And then we have what's called roan, which is big R, big W. Oh, that's a W. So roan is patches of the reddish brown color on a white coat. All right, so it's patches. So the fact that the red is big R, the white is big W, and the roan is big R, big W, let's see what the question asks you. Do either of these alleles exhibit dominance over the other allele? No, right? Because we have red, we have white, and we have roan, which is, reddish brown and white patches. So we see both phenotypes here. So then what is the name given to this type of allelic interaction? Codominance, right? Why is it not incomplete dominance? But why is this not incomplete dominance?
They don't blend together or exhibited separately.
Right, so both colors are in the phenotype, so we see both colors, right? It's not an intermediate color. And also the way the question is written with big R, big W, right? Big R, big W, you know that it's not a case of incomplete dominance. So it's actually codominance. Both alleles are being expressed equally, and both phenotypes are observed. So it's not an intermediate phenotype, a pink cow, right? So it is an example of codominance. So then we have some crosses. Our first cross is a red and a roan. So a red with a roan. And you could simplify this. I'm not doing that this time, but you could. So our phenotypic ratio is 1:1, red and roan. Oops, sorry. Red and roan. You could have said 50-50, right? That works too. Our genotypic is also 1:1, big R, and oops, big R, big W. Or you could have said 50-50, right? That's fine. Or half and half. The next cross is a roan and a roan. A roan and a roan. And here we get everything, don't we? So our phenotypic is 1:1, right? Red, roan, and white. It's not 3:1. But aren't we crossing two hybrids? RW with RW? I mean, they're both hybrids. Why do we not get a 3:1 phenotypic ratio?
Because of codominance.
Right, because it's a codominant situation, right? So the codominance has changed that ratio to a 1 to 2 to 1 ratio, and it's the same ratio for the genotype as well, right? It's 1 to 2 to 1. Or you could say 25%, 50%, 25%, right? There are different ways to express these numbers. The next cross is a roan and a white. So roan with a white. Again, you can simplify, but I'm not doing it here. Oops. So half the offspring are roan and the other half are white. So our phenotypic ratio is 1 to 1, roan right to white, or you could say 50-50. It's the same for the genotypic ratio, right? It's also 1 to 1. RW to WW, right? Or you could say 50-50, a half and a half. And then our last cross is a red and a white. You don't even have to make a square, do you? What are all the offspring going to be? They're all roan, right? 100% roan. And that's the same for the genotypic or phenotypic frequency or ratio. 100% roan, 100% RW. So this is a good problem to illustrate codominance. It shows how the ratios change, right? Because it's a codominant situation, right? So a lot of our ratios are 1 to 1 or 1 to 2 to 1. So any questions on the codominance problem?
Yes, you said that the offspring would be 100% wrong. Can you please explain that again?
Well, because the red animal can only donate a big R allele, and the white can only donate a big W. So all the offspring are going to be big R, big W. They're all going to be roan.
Got it. Thank you.
Crystal, for the sake of consistency, right, so that we know, I mean, in reality, RW equals WR. But just to be consistent, we're putting the flower color first, or I'm sorry, not excuse, we're putting the roan, the red color first, the big R allele first, and then the big W allele after, just to be consistent. Right, because we're told in the problem where it says roan, it specifically writes it as big R, big W. And so we're just going to be consistent and keep the R's first, and then followed by the W's. So when you do these problems, it's important to be consistent. So again, whatever the problem tells you, follow the pattern that's given to you in the problem. Otherwise, it gets really confusing, especially when you do a dihybrid cross, which we do have a dihybrid in here. And so we have to be consistent, right, with how we write things down. So horses, right, problem for horses. So here we're going to get into one of those dihybrid crosses. So horses, black coat color is determined by a dominant allele, big B, Well, the recessive allele determines the chestnut coat color, so a brown coat color. So let's write down what we're given. So we know the big B allele is going to be for black coat color. It's dominant to little b, which gives us our chestnut. And because we know that big B is dominant to little b, you can be big B, or big B, little b, and have a black coat color, but you have to be homozygous recessive to have the chestnut coat color, right? So this is all stuff that we know from reading the problem. The other characteristic has to do with how the animal moves. So big T is for a trotting gait. So big T we're going to call trotter, right? The horse can trot. And then a little t is a pacing gait. And so a little t is recessive. So we know that our big T is for trotter. Our little t is for pacer. Again, big T is dominant to little t. So we know if you're big T, or big T, little t, you have the trotting characteristic. If you're little t, then your horse is a pacer. So now we've got two characteristics, right, to deal with. We're going to put the B's first, followed by the T's, because that's how the problem states it, right? They gave us the coat color first, followed by how the animal moves, trotter or pacer. And then our first problem is to determine the genotype and phenotype of F1 horses, right? So the first generation of horses derived from a cross between a homozygous black pacer and a chestnut homozygous trotter. So a homozygous black pacer. So we know it's homozygous black. And it was a pacer. So we know that being a pacer is homozygous recessive. And we're going to cross that individual with a chestnut homozygous trotter. So it's chestnut, has to be little b, and it's a homozygous trotter, big T, big T. Right, so this is what we're being told in the problem. Now I am going to simplify here because we got a lot of stuff to do, but this individual, all of its gametes are big B and little t. This individual, all the gametes are little b, big T. So we don't have to write it out 4 times. All the offspring are going to be dihybrids. They're all going to be black trotters. So now you can guess what the second cross is going to be, right? So these are the F1s. Our second cross is going to be we're going to cross 2 of these F1s. So we're going to have a good old-fashioned brother-sister mating. So since the F1s are both dihybrids, we have ourselves a big dihybrid cross. So this is one of the 4x4 crosses. So it's one of the big ones. It's going to ask us for all the phenotypes that occur. So if we do our FOIL technique, big B consort with big T, big B consort with little t, little b consort with big T, and little b with little d. Since we're crossing two of the exact same genotypes, all the gametes are going to be same for the other horse. So we got a big cross. And here's where we have to be careful that we be consistent with how we write things down. So B's come before T's. So big B, big B, big T, right? And I'm going to keep track of these over here. Next one, big B, big B. That's a B. Big T, little t. Everybody follow what I'm doing? So next row, big B, big B, big T, little t. We already have that, don't we? Here, so we had it once, now we have it again. So I'll just put a mark here for each time we see it. Big B, big B, little t, little t. Big B, little b, big T, little t. We already have that one, right? Big B, little t, little t. And just one of those so far. Big B, little b, big T. So we already have that here, so we'll mark it down again. Big B, little b, big T, little t. Yeah, we got that. Little b, big T. All right, that's a new one. Got one of those. Little b, big T, little t. So we already, I don't think we have one of those. Big B, big T, little t. Yeah, we got that. Big B, little t. So big B, little t. We got that here. Little b, big T, little t. Yeah, we have that one. Another one. And lastly, so that's one of those. So those are all the possible genotypes that occur in that table. So let's see, what do these look like? Big B, big T is a black trotter. Big B, big T, little t is another black trotter. Big B, big T is another black trotter. And big B, big T, little t is another black trotter, right? Those are all black trotters. How many black trotters are there?
Four.
Hmm?
9.
9, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 black trotters. So those first 4 genotypes are all the same phenotype. They're all black trotters. Our next one is black pacer. Okay, that looks bad, but a black pacer. Big B, little t is another black pacer. How many black pacers?
3.
3. Our next phenotype is going to be a chestnut trotter. And then a little b, big T, little t, again, is a chestnut trotter. How many chestnut trotters? 3. And the last one, all by itself, is a chestnut pacer. And there's only one of those. Does this phenotypic ratio look familiar to you? Whenever you do a dihybrid cross, right, where both individuals are hybrids for 2 characteristics, you'll always get a phenotypic ratio of 9 to 3 to 3 to 1. That is characteristic for a dihybrid cross. So what I did was just, as I was filling in the table, I wrote the unique genotypes, and then I marked out how many of them there were. That's why we got the big B, big B, big T, big T, big B, big B, big T, little t, and so on and so forth. All the unique phenotypes. And then how many times they occurred in that table. And then I wrote out the phenotypes for all those individuals, and then added up how many of them there were. There are 9 black trotters, 3 black pacers, 3 chestnut trotters, and 1 chestnut pacer. 9 to 3 to 3 to 1 is our phenotypic ratio. And the reason why they never ask you for a genotypic ratio is because that doesn't mean anything. You say like a 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. That doesn't mean anything in this case. So all they ever ask is about the phenotypes in a dihybrid cross. So questions?
Professor?
Yep.
Yeah, so I get everything except for how you spread out the BT to make the table.
I couldn't hear that last part.
I said how did you can you explain how you spread out the BT, right, to make the table?
Oh, the FOIL technique here.
Yes.
Right.
Like
So
I know how we filled up the table, but how we, placed the B's and T's. Can you please explain that again?
I mean, how we get all the gametes on each side here, right? So in this case, with a dihybrid, we can do that FOIL technique. And so remember, because of the laws of independent assortment and segregation, we're never going to get a case where we got 2 B's together in an egg or sperm, or 2 T's together in an egg or sperm. So we're going to have a big B, it can sort with the big T in an egg or sperm. And the big B can sort with a little t in an egg or sperm. And little b with a big T in the egg or sperm, or little b with little t in the egg or sperm. We can never get, right, that never happens. That never happens unless it's a mistake, right? A non-disjunction or something like that. with the chromosomes and the chromatids, but that's, not something that's going to be viable, right? The offspring are not going to survive. So we only include those probabilities that are most likely to occur. So does that help explain?
Yes.
All right, so the next problem I like because it's a deductive reasoning type problem. So in the next problem, we're going to be dealing with squash plants and two characteristics, the color of the squash and the shape of the squash. And so we have a white color squash, which is dominant to yellow color. So we're going to be using W's, a big W for the white color and a little w for the yellow color. So that's one characteristic. So again, let's write down what we know. We know that big W, oops, sorry, I keep doing that. A big W is dominant to little w. So we know that big W is a white color squash and a little w is a yellow color squash. So because big W is dominant to little w, we know that you can be homozygous dominant or heterozygous and have a white squash. But to have a yellow squash, you need two little w's, right? It's a homozygous recessive condition. The other characteristic is the shape. We have disc-shaped squash is dominant to sphere-shaped squash, right? So disc-shaped squash, big D, is dominant to sphere. Sorry, sphere-shaped. So you have to be big D, or big D, little d, and you can be disc-shaped. But you got to be homozygous recessive to have a sphere shape. So that's all the information that we're given in the problem. So we have several crosses. The first cross is a white disc with a white disc. And we're being given all the offspring and the numbers of the offspring. So white disc, if we have a white disc with a white disc, we know that they have to at least have a big W allele in order to be white and a big D allele in order to be disc-shaped. And we're crossing two of these individuals. Now I'm writing it like this because all we know is that they are white color and disc-shaped. We know that white is dominant, so if you have a big W allele, then the squash will be white. With disc shape, disc is dominant to sphere. And since it's a white disc, we know it has at least a big D allele. We don't know what the other alleles are yet, right? Because all we know, it's a white disc. It could be homozygous, right, for each characteristic. It could be heterozygous for each characteristic, or it could be a mix. Heterozygous for one characteristic, homozygous for their characteristic. We don't know yet. That's what we're trying to figure out. What are the genotypes of the parents, right, that produce the offspring? So what kind of offspring do we get? Well, we actually get basically everything. White disc, white sphere, yellow disc, and yellow sphere. So we get white disc offspring. We get yellow disc, or sorry, white disc, white sphere next. So white sphere, so again, we know it's that, and sphere shape, well now we know it's got to have two little d alleles to be sphere shaped. The other offspring, yellow disc, well, yellow, we know it's got to be two little w's to be yellow, and it's a yellow disc, so we know it has to have at least a big D allele. And then lastly, we have white spheres. I'm sorry, yellow spheres. All right, so yellow sphere we know is homozygous recessive for both characteristics because that's the only way to get yellow and the only way to get sphere. So we know these individuals have two little w's and two little d's. So there's actually more than one way to solve this problem. One way is by looking at the possible genotypes that I've written here. And so this is the key. We have offspring that are homozygous recessive for both characteristics. So remember that recessive allele has to come from both parents. In order to express the recessive characteristics, you have to get the recessive allele from both parents. So both parents are carrying the recessive alleles. So well, then we already know that we have to have little w and little d and little w and little d, because these four alleles end up in this offspring. So they have to come from both parents, right? Since both parents are expressing the dominant characteristics, we know they're carrying the recessive alleles, right? We don't see them, right? Because they have the dominant alleles. We only see the dominant characteristic. But some of their offspring have both recessive characteristics. So those had to come from the parents. So that's one way, right, to do that problem. And the other way is to look at the numbers, right, that were given. So they said 29, 10, 9, and 2. So 29, 10, 9, and 2. Do you see anything special about those numbers? Yeah, right. It's roughly 9 to 3 to 3 to 1. So we have a 9 to 3 to 3 to 1 phenotypic ratio. That means both parents have to be dihybrids in order to get a 9 to 3 to 3 to 1 ratio. Yes, the numbers are not perfect because they rarely are. Right? The only way to get perfect numbers is to have thousands of offspring, and then the numbers will come out perfectly, right? Because you have so many offspring. But we didn't have that many offspring here, right? So it's roughly 9 to 3 to 3 to 1. So there's two ways to solve that first problem. Look at the possible genotypes of the offspring, and we find there are recessive alleles. And of course, in this individual here, we got two little w's, and this one, we got two little d's. And the only way to get those recessive alleles, they have to come from both parents. So if you find the recessive characteristic in the offspring, that means the parents are carrying that recessive allele. So questions on that? So next we have a white disk and a white sphere. So again, white disk has to be that. White sphere is that, right? So we know that this parent is homozygous recessive because it's sphere-shaped. We don't know about any of the other characteristics for sure, except that they're carrying a dominant allele. So we look at the offspring. In the offspring, we have white disk and yellow disk. So we're going to get white disk and yellow disk. Do we have any white spheres? Any sphere-shaped offspring? No, there's no sphere-shaped offspring. So that tells us something. right? If this parent here had a little d, we would see little d in the offspring, right? So we'd have some sphere-shaped offspring, but we don't see that. So there can't be a recessive allele there. It has to be a dominant allele. Otherwise, we would see sphere-shaped offspring. We don't have any. They're all disc-shaped. We do have some yellow offspring, don't we? Here we got the yellow offspring. So it has to get make that bigger. It has to get the two recessives, one from each parent. So that means that this parent is carrying it and this one is carrying it. That's how we get it in the offspring. Now the numbers here don't really mean much, except that you can confirm them if you do a cross, right? So we have a 3/4 and 1/4, right? This is a this is a cross involving two different characteristics. So it's not a monohybrid cross. So that 3:1 ratio, that doesn't apply here. But if you were to do this cross now, right, this individual, big W, consort with, big D. It'll happen twice, but I'm just going to show it once, just for simplicity. And the little w consort with a big D. So I don't have to write big W, big D, big W, big D, little w, big D, little w, big D, because I don't have to duplicate, and I don't have the room to do that. So we're going to simplify. And this individual here, a big W consort with a little d. Or a little w consort with a little d. Again, yes, there are two of each gamete, but I don't have to show them because I don't have to duplicate my efforts. Oops, two little w's here. So the first individual, or the first squash plant here, big W, big W, big D, little d, what does it look like? It's a white disc. Big W, big D, little d, what does that look like? White disc. Big W, big D, little d, again, white disc. Our last one here, yellow disc, and how many? One. So we got 3 out of 4 white disc, 1 out of 4 yellow disc. So with these problems, the squash problems, especially when you go back and do them, do the crosses for practice, right? So once you have an idea, or once you know what the genotypes of the parents are, go and do those actual crosses and then see what the offspring are. And they should all match what's in the problem. So any questions on that one? Because now I have to blank this to go to the next one. Oh, which is still squash, so we're still doing squash. But this time we're going to cross a white disc and a yellow sphere. So again, white disc We know that. Yellow sphere, we know that's yellow, and we know that's sphere, right? So we actually know the complete genotype of one of the parents. It's yellow sphere. And it tells us one half of the offspring disk and the other half are white sphere. So we've got white disk and white sphere. Right, so it's telling us in the offspring, half are white disk, half are white sphere. Do we have any yellow offspring? No, right? Because yellow would be little w, little w. We don't get it. So then we know this parent has to be homozygous dominant. If it were heterozygous, we get two little w's in the offspring, and we'd have yellow, right? But we don't get that. And then we do have sphere, right? We've got the sphere. Each recessive allele has to come from or has to come from each parent, right? So that tells us that this parent has to be carrying the recessive allele. And that's how we get two little d's in the offspring. So, and we're told that this is half and half. If we were to do the cross, big W with big D, big W consort with little d, and this is just, a repeat, right? So we're not going to duplicate. And this individual, all little w, little d. You don't have to write it 4 times. So big W, big D, little d. Big W, little d, little d. 50-50, right? Now we know this one is big W, big D, little d, and little w. 50-50, half and half, or 1-to-1. Questions? And in the last squash problem, white disk, white sphere. So white disk, sorry, white disk, crossing it with a white sphere. And what kinds of offspring do we get? We get white disk, white sphere, yellow disk, yellow sphere, right? So we get white disk, white sphere, white sphere, and then we get yellow disk and yellow sphere. Yellow sphere. Do we see the recessive traits in the offspring? We especially do here, right? But we also have it here and here. Where did those recessive alleles come from? Each parent. And so because we find our recessive alleles in each parent, that's how we know the genotypes for those parents. Now here the numbers we're given are 38381818. 38381818. So 3311, not 9-3 to 3-1, but 3311. So that doesn't tell us anything special. But if you did the cross, right? So Again, we have a dihybrid here, right? And this individual here, we have big W, little d, little w, little d. We don't have to duplicate because this just keeps it simpler. Big W, big W, big D, little d, big W, little w, big D, little d, big W, little d, little d, big W, little w, little d, little d, big W, little w, big D, little w, big D, little d, oops, big W, little d, little d, little w, little w, little d, little d. So big W, big W, big D, little d is a white disk, right? We got a white disk. Big W, little w, big D, little d is also a white disk. Big W, little d is a white sphere. Big W, little d, little d is white sphere. Big W, big D, little d, white disk. Little w, big D, little d, yellow sphere, yellow disk. Big W, little d, white sphere. And little w, little d is our yellow sphere. 3, 3, 1, 1. We have 8 options in the table, right? 8 possible offspring. So 3 out of 8, 3 out of 8, 1 out of 8, 1 out of 8. Questions? So the key to the squash problems, write down what you know. Write down what you know about the genotypes of the parents, then write out the genotypes of the offspring based on what you know and see what's missing. Once again, if you find the recessive characteristic in the offspring, that means that both parents are carrying the recessive allele, right? Because the only way for the recessive trait to appear in the offspring is the offspring are homozygous recessive, which means each recessive allele has to come from each parent. Any questions about squash? Our next problem is a X-linked or sex-linked problem, but since we're only talking about the X, all sex-linked problems are X-linked. So we have to show X's and Y's, right? So we need to know the gender of the offspring when we do our crosses. So we're talking about hemophilia, which we've talked about, bleeder's disease. They do not have clotting factor, so they can't clot. And so they bleed a lot, often to death. But this gene is X-linked. And the gene for hemophilia is recessive, right, to the allele for normal clotting. So we're going to use big H and little h. But remember, we're going to put these on the X chromosome. So a big H is normal clotting, and a little h is hemophilia. So we know a big H, little h is normal, and little h would be hemophilia, except that we're putting these on the X. So we have to show X's and Y's, right? We have to show XX for females, XY for males. There is no complementary allele on the Y chromosome, right? Because we know that males are hemizygous. So our first cross is a homozygous normal female and a hemophiliac male. So homozygous normal female, big H, big H. And we're going to cross that with a poor hemophiliac male, right? He's only got one X. It's got the recessive allele. He expressed that characteristic because there's nothing on the Y, right, to complement the X. But the female is homozygous normal, right? She's homozygous normal. So we set up our cross. Oops, that's a Y. So now we're looking at males and females. The symbol for females is this, and the symbol for males is that, right? So that's how we write the symbols for male and female. So for the females, right, that's the only genotype for the females. And for the males, only one genotype for the males, right? So all the females are 100% normal. All the males are 100% normal. But the females are carriers, right? They're carriers for that recessive allele. But all the females will have normal clotting, right? Because they have one normal allele. So if you're heterozygous, you're normal, right, for clotting. In this case, all the males will receive the normal allele. So all the males are all good. So for the next problem, you can guess it's going to be different, right? The next problem, we're going to cross a normal female and a heterozygous female. So we're actually going to cross these two individuals. Hopefully it's not a brother and a sister. But now we're going to cross a heterozygous female with a normal male. So the gametes from the female, half are carrying the good allele, half are carrying the bad. And for the male, all his X's are good. He's got a Y. So X big H, X big H, X big H, Y, X big H, X little h, X little h, Y. Uh-oh, that guy's in trouble. So for the females, they're all normal, but half are carriers, right? Because they're heterozygous, but they're all normal, right? Phenotypically. But half of them are heterozygous, so they're carrying, right, that recessive allele for hemophilia. When it comes to the males, well, half the males are lucky, right? They're normal, but the other half have hemophilia. So for the males, it's a 1-to-1 or a 50-50, normal to hemophiliac. Because if a male inherits that defective allele, he has hemophilia because he's hemizygous, right? There's nothing on the Y chromosome that's going to help him. But females can be heterozygous. One normal allele, one defective allele, and they're okay, right? They'll be able to make enough clotting factor in order for the blood to clot normally. But whenever you have an X-linked or sex-linked trait, you must show the allele on the X chromosome. And you must show X's and Y's, but the allele only appears on the X chromosome. So questions on that? Are we good? Does anybody know of any other X-linked traits that men suffer from?
Is color blindness one of them?
I think so. But I was thinking of one that affects me, which is baldness. All right, so the most common male pattern baldness is X-linked. So all those bald guys out there, it's mom's fault. Because he got it from mom. And that's why women who carry that trait, they normally don't start to express it until after menopause. Because after menopause, their estrogen goes way down. And what little testosterone they have now starts to exert its effect and cause hair loss. So that's why older women tend to have thinning hair, because they're carrying the gene, right, for baldness. It's just that you need testosterone to really express that gene. So us guys with a lot of testosterone, that's why we lose our hair. And so women don't have that much to begin with. and it really can't exert much of an effect while she's producing a lot of estrogen. But after menopause, estrogen goes way down, so now testosterone can start to exert its effect. And so that's one of the reasons why many older women lose their hair, or a female bodybuilder who is carrying the gene for baldness, if she uses steroids, her hair starts to fall out. And once it starts, you can stop the steroids, no more hair will fall out, but no new hair will regrow. So, and I've seen that. So women who use steroids try and bulk up and their hair started falling out. So, and they stopped and the hair loss stopped, but what fell out did not regrow. So our last problem, and we'll have time to do the last problem, so that's good. So here we're dealing with poultry, so like chickens and roosters, particularly the roosters, they have this little flap of skin above their beak, in between their eyes, and there are different shapes for that skin flap, that growth that's there. You don't need to know what they look like, but they have what are called walnut, rose, pea, and singles for their combs, right? That flap of skin that's up there above the beak. And it actually does serve a purpose in poultry. The hens are turned on by different kinds of combs. So, but anyway, so let's write down what we're told here. So walnut, and we're using R and P for the alleles. I'll just reproduce what's there. So we got equals, so walnut. Big R, 2 little p's. And that was what? Rose. Two little R's, and then what was it? And a big P is the P shape. And lastly, we have our single, which is homozygous recessive. So that's what we're told. If you have those combinations of alleles, those are the type of comb that you have. So we're going to cross a big R, little r, big P, little p. With a big R, little p, little p. Right, so a walnut and a rose. So we can do our FOIL technique to get the gametes, right? Big R consort with big P. Big R consort with little p. Little r consort with big P. And a little r consort with little p. With this individual, a big R consort with a little p. Only going to do it once, don't need to do it twice. And a little r consort with a little p. Only going to do it once, don't need to write it twice. Right, because we're going to get the same ratios at the end. So big R, big P, little p. Big R, big P, little p. Big R, little p, big R, little p, big R, big P, little r, big P, little p, big R, little p, and little r, little p. So if you have a big R and a big P, you're a walnut, right? So a big R and a big P is a walnut. So here we have a big R and a big P, a big R and a big P, a big R and a big P. So we got 3 of those. That's our walnut. Rose, we know we have to have a big R and 2 little p's. So big R and 2 little p's here, big R and 2 little p's here, big R and 2 little p's here. A P has to have 2 little r's and a big P. Here's 2 little r's and a big P. Got 1. That's it. And then for single, gotta have 2 little r's and 2 little p, which is right here. That's our 1. So a 3, 3, 1 to 1, or 3, 8, 3, 8, 1, 8, 1, 8 is our ratio, right, for the phenotypes. So questions? You guys didn't say much, so we got through all the problems. Usually that doesn't happen. So now that you've had a chance for me to go through them, now you can go through the problems and try them all by yourself. And if you get stuck, then you can refer back to this recording, right? Because all the answers are there in order. So questions?